Well, good morning, everyone. Bonjour tout le monde. Bienvenue. Welcome online and in person to Concordia University's Fourth Space. Thank you so much for joining us for Research and Conversation in the Faculty of Arts and Science. To help get you situated, we are streaming to YouTube live from Fourth Space, which is located on unceded Indigenous lands here in Jogé, Montreal. And we'd like to extend our gratitude to the Kanien'kehá:ka Nation, who are the caretakers for the lands and waters we're meeting on today, for their teachings about the earth and our relations. If you're new to Fourth Space, welcome once again, online or in person. We work with our university community here in order to mobilize and exchange knowledge by creating daily ways and formats for us to be able to do so. Podcast conversations such as today's, roundtables, workshops, hackathons, etc. We're so very pleased to welcome back Associate Dean of Research Patrick Leroux into Fourth Space and his special guest, Claudine Gauthier, here this morning as they embark on a conversation about Claudine's work, impact of aging and lifestyle on the brain. We're so excited. We've got such a packed day that I'm going to pass the floor to Patrick now. Welcome.
Thank you, Anna. This is great. Thanks for having us yet again. We've been meeting with researchers in the Faculty of Arts and Science over the course of the year. And today we actually have three colleagues coming in. Claudine Gauthier, Associate Professor of Physics, who I'll start my conversation with, followed by Tianming Zhang, an assistant professor, in chemistry and biochemistry, and finally Marek Majowski, an assistant professor in chemistry and biochemistry as well. So it's a day of science. I'm not necessarily in my comfort zone, which is actually pretty exciting. I mean, literature, obviously, and performing arts, and I haven't really done or thought about chemistry, physics, biology since 1987. So this is, nothing's changed, right, since the 80s?
No.
Right. Claudine Gauthier, it's truly a pleasure having you here today. We've had exchanges over the past while on various topics, but this is an opportunity for us to really dive into your research. I'll summarize. So Dr. Gauthier completed her PhD in investigating cerebral physiology and healthy aging using quantitative fMRI, so functional magnetic resonance imaging, at the University of Montreal. And after a PhD, Dr. Gauthier did a postdoctoral fellowship in neurophysics and neurology at the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig, Germany. She joined Concordia as an assistant professor in 2014 and became associate professor in the Department of Physics in 2019. I'd like to point out also that she holds the Henry J.M. Barnett New Investigator Award from the Heart and Stroke Foundation of Canada, and extremely importantly, I think, is the Horstein Chair holder of the of cardiovascular imaging at the Montreal Heart Institute. So professor at Concordia in physics,
yes,
chair at the Heart Institute. You've done, you've studied in biochemistry, in neurology, neurological sciences. What is your trajectory? How did you end up in physics, going through all these various permutations of scientific inquiry? Tell us a bit about that, please.
Well, I started in biochemistry, and my theory about this is my entire family is engineers, and I was trying to get as far away as engineering as I could, and then I slowly drifted back towards, my roots. And so I started in biochemistry, and then in my master's degree at McGill, also in biochemistry. But I actually found that biochemistry was it's very manual. To say you have to, manipulate little tubes, and I'm kind of distracted and often a little bit clumsy. So I would often have trouble with my experiments, spill it, miss my timing. I don't know. I wasn't very good at it. And I had some friends who were doing imaging at the Montreal Neurological Institute, and I would have lunch with them, and I thought, oh my God, these people, they look so happy doing what they do, and I want to do that, right? And I think one of the reasons why they were so happy doing it is that imaging is a very new field, especially then. And so it's very collaborative, right? And I think that's kind of what I wanted, something that was very collaborative. And so I moved on to imaging and never looked back. And in imaging, then I kind of did a lot of more kind of technical development during my PhD and my postdoc, which was the kind of physics aspect. and then did some application in physiology because that was kind of closer to my original interests in starting university. And then in the end, kind of combined them together, and yeah, got hired at in physics to try to develop the kind of medical physics part of the program.
Yeah, because the medical aspect and the basically your study in lifestyle choices, and we'll get to that in a moment, also open doors to Engage. So you're involved in Engage. You're also involved with the AI Institute, the Artificial Intelligence Institute. I think it's the cluster on artificial intelligence and science. Could you tell us, I guess, what is the fundamental focus? If you had to summarize, like the one theme, the Christmas dinner party, one line description, and then we can dig in.
Yeah. So imaging is a very versatile tool, but it's often used in non-quantitative ways. And I think the problem with that is that you can't compare different people that may have disease together if you do that. And so really the focus of my research is to try to develop and use quantitative techniques using MRI to study aging, disease, lifestyle, right? But really focusing on this quantitative aspect of it.
Right, and the quantitative aspect is part lab work, so controlled lab experiments, but also I understand you also have students looking into publicly available data.
Yeah.
How does that work, and how do these data sets compare, and how do they complement each other?
Yeah, so publicly available data sets tend to be very large data sets, but they often don't have a lot of quantitative information in them because those are much harder to acquire, and often they require special manipulations and things like that. And then the data that we acquire in the lab tends to be in these more complex experiments, smaller samples, but more quantitative, right? And usually students will kind of have two projects, one of them working on these publicly available data sets where the numbers work for you, and then in data that we acquire ourselves where really you don't have a lot of people, but it's much more quantitative, and they kind of get a taste of both types of data, which I think are important for the future. Like these big data sets are definitely a big part of the future of science. So it's important for them to learn how to work with that data as well.
Right, and are you expected to contribute your own data into those large data sets afterwards, or do you keep those separate?
Yeah, those are separate. I mean, definitely open science is a really big direction in which everybody's pushing these days. And so I think there is an expectation that the data that we acquire will be available to the community in the future. I mean, we're not finished acquiring it, but in the future to make it available to other scientists. Yeah. But those tend to be separate things. They each have their own protocol, which is different, so they can't really be mixed together. But definitely, it is expected now that scientists share their data with the scientific community.
Right. So let's step back a tiny bit, and so the data sets you're creating, the experiments you're working on, the various fMRI captions, essentially you're capturing. Who are you basically studying? Because you're looking at aging, you're looking at cardiovascular issues, and you're also looking at lifestyle. So could you tell us a tiny bit? Who are you measuring? What are you looking for? How is this done? And are you looking across the lifespan? Or are you focusing on the elder population?
Yeah, so my plan eventually is to have this type of data, this very quantitative data that not many people have, on a very wide range of people across the lifestyle and different disease spectrums.
Right.
But it's difficult, it takes time, it takes a lot of money to do that. So basically we're kind of filling pieces of the puzzle as we go along. And right now we have three studies going on, one of them on coronary heart disease, one on long COVID, and one on the menopause transition.
Okay.
And each of these, for example, long COVID and coronary artery disease, we have a patient population, but we always have a control population also, right? So we're basically kind of filling the puzzle both in terms of patients, but also always healthy people. And in many ways, I mean, the healthy people is kind of the database you want to grow the most, because if you want to look at subtle effects of lifestyle, for example, or hormones or things like that, then you need quite a big number of people, right? So by kind of combining people from multiple studies, eventually we have this large data set that we can look at these effects in.
Right. And in terms of the population, I noticed in one of your many grants that you were looking at the difference also in men and women in certain activity. Can you tell us about that? Like, you've noticed differences, right?
Yes.
Yeah.
So this is CIHR has pushed a lot in recent years to really get people to look at sex and gender, but I think people are starting with sex mostly in much more detail than before. So really CIHR has a whole guideline on how to do sex and gender-based analysis. And it really involves kind of separating men and women, well, males and females really, and studying those two populations separately, because the biology might be different in the two sexes, right, for many different reasons. And when we started doing this with our old data, we realized that a lot of things that didn't make sense, relationships between, I don't know, arterial stiffness and brain health and things like that, they didn't make sense. And the reason why they didn't make sense is because we were combining everybody together. And once we separated males and females, we could see relationships that we had never seen before. Because if you're putting them together and you're taking the effect of sex into account in your statistics, what you're looking for is what is the mechanism in common between them, right?
Right.
And the mechanism in common between them might not actually be the interesting mechanism for development of disease. Sometimes the pathophysiological mechanism in each subgroup is actually the relevant one, right? And when we started dividing people, everything makes so much more sense all of a sudden that now we've really heavily invested in that to try to understand how aging happens in males and females, and a lot related to exercise recently, especially.
Okay, can you give us an example in terms of
exercise? So for example, we've been looking at data on exercise intensity and how it relates to brain health in males and females in middle age and old age. And what we've been finding is that, for example, in males, to get a benefit in terms of brain health of exercise, males need relatively high intensity of exercise. Low exercise intensity doesn't really do that much for them, but if they do more high intensity exercise, they get more benefit. Females, on the other hand, are completely different. So vigorous exercise doesn't seem to make that much difference, but a total dose of exercise, and especially walking, is very beneficial. So that, you know, the recommendations right now, which are really based on this kind of more intense exercise, are really appropriate for males, but not so much for females. It's not that it's not beneficial for them, but really, I think guidelines towards for females in our data really show that it should be more geared towards walking and total dose of exercise, no matter what type of exercise it is.
Okay, and is this true throughout their entire lifetimes, or is this a specific population you're looking at? So
for males, it's true in, I mean, the data only starts in people in their 30s. So in males, it's true throughout that later years lifespan. So basically middle and older age. In females, on the other hand, it's mostly true in older females. So younger females who are not menopaused yet don't really seem to derive a benefit from exercise in our data, though I really need to emphasize that this seems to be due to the presence of estrogen. So probably what happens is estrogen, because it's so protective, basically means that you really don't see the effect of exercise right then, but it probably has a beneficial effect on later life, right? So basically estrogen would mask any effect of exercise while it's still there. But if you do exercise, then in later life, you would be healthier, right? But we don't see it right away until menopause, basically. Right.
And how will we see this? How do you measure this? So you work with imaging, brain and vascular imaging. So you're looking at very specific moments.
Yeah.
how would you anticipate these benefits, the estrogen-related benefits in the long run?
Yeah, so probably it has to do with vascular structure, right? So we know that blood vessels become harder in aging. So basically, blood vessels in their walls, they have a protein that's called elastin. And elastin is called like that because it's basically like elastic. It can be stretched. And this is what makes blood vessels compliant, so they can increase their diameter in response to a bolus of blood, basically, that comes from the heart. And just like if you stretch an elastic many times, it'll start to be stiffer because the proteins inside that are elastic break. The same thing happens with elastin. So as we have heartbeat throughout our lives, the elastin protein breaks down and it's not replaced. And so the more we age, the more our vessels become stiff. And we know that exercise and other positive lifestyle experiences will tend to make this process happen less. So there will be less breaking of these elastic proteins, and so the vessels stay healthier. And so it's probably something related to this, or formation of plaques in vessels and things like that.
Well, as you describe this, I can feel my own vessels hardening and thinking about what we should be doing. I'd like to understand who your principal interlocutors are. So you're working in your lab, but you're also working really across different institutes, Engage AI, as we mentioned, and also with the Heart Institute as well. Who does your science speak to? And how is it received? And I'd like to understand like that connection, basically.
Yeah, so most of my research so far has really been targeted at, basic science, in a sense that we can understand mechanism of aging and disease, other scientists, and also clinicians. So a lot of the techniques that we're developing, they're potential biomarkers, we call. So they're potential markers of a physiological change that could be kind of early in the disease process. So the idea is we develop these techniques, we make them accessible, and then you hope that a clinician, multiple clinicians, will adopt them into their own practice and acquire this data in their patients so that we can basically follow the development of disease and maybe either help in diagnosis of disease or help in therapy. Because if it's a biomarker that is sensitive to changes in lifestyle or drugs, for example, then we can use it to follow the course of therapy. So that's the main stakeholders. And then this exercise branch that we're exploring now also has policy implications, as I mentioned, because a lot of what we're finding is that basically the guidelines that exist, they're very broad, and there's nothing wrong with them in the sense that if you follow them, for sure you will be healthier, but that there may be benefit in terms of adoption and people actually following these guidelines if they were made more appropriate and different for different age groups or different sexes or genders.
You're in physics, you're in Engage, but you're also a chairholder at the Montreal Heart Institute. What does this mean? And what does this give you in terms of access, resources? Does it bring you out of Concordia, or does it bring resources here?
So, I mean, it's mostly what it gives me is, funds to do research, which is always nice. But also access to patients, because Concordia doesn't have a medical school, so access to patients is a little bit more complicated here. And really, because I'm looking at vascular, metabolic health and aging, and we're basically trying to develop these biomarkers. The way this is done usually is we start with disease populations to see if our biomarkers can pick up the effect of disease, and then we can dial back the disease basically towards a normal population to see how much of the the variance in the population we can pick up. So, but to be able to do this process of figuring out how sensitive our biomarkers are to relevant physiology and the development of disease, we need patients, right? And those patients are at the Heart Institute, so that allows me to have access to them.
And concretely, what does this mean? Do you spend X number of days per week at the Heart Institute? Do you ask permission for a patient to come over and look at their fMRI? Or I'm trying to understand how you do this.
So basically, it means I spend I spend some time every week there, but my students spend a lot more time there because they're the ones who do the data acquisition and all that. And so a lot of our projects, not all of them, but a lot of our projects take place there. So that's where we do all the data acquisition, and then we have all our computers and all our data analysis infrastructure at the physics department, and then so they kind of go back and forth, which, you know, poor them, because those two are kind of at the other, each end of the city there. And so yeah, they go do data acquisition a few times a week at the Heart Institute, and then they come back to analyze the data at Concordia, basically.
Right. And is it helpful to be on site at the hospital to have a sense of who it is whose data you're looking at? Is that taken into account in the analysis, or do you keep that absolutely separate?
I mean, I think the presence there is good for, I mean, aside from that's where we analyze the data, I think it's also a community that's very, you know, knowledgeable in terms of vascular disease and physiology of, you know, vascular physiology and all that. So we get a lot of kind of scientific effervescence from being there, right? Yeah. And then, but definitely when we try to analyze the data, you know, it's very important not to be biased when we analyze the data, because we always have expectations about what we want to find, right? You know what you want in that nature paper, you know? So I think it's really important not to think too much about who you're analyzing when you're analyzing the data, because you don't want to be biased. You know, oh, you know, I think that's an artifact in this data because, you know, it really doesn't go in the direction I expect, so it must be something wrong with my data, right? Things like that. So it's really important, I think, to keep as unbiased a view of the data as possible. And
how closely do you work with the treating doctors, for instance, on site? Are they involved in pointing out certain interesting cases? Do they want some feedback on what you found pretty quickly, or is it pretty hands-off?
Well, so they help us with recruitment. So usually, especially, for example, the coronary artery disease patients that we're doing now. So we get them from doctors who are treating them at the Montreal Heart Institute. So we'll, you know, go through their patient files. I mean, my students do this. I don't do this. And, you know, sit with the treating physician to, you know, see if they fit our criteria, if we think they would be a good case for us. And then, of course, once it's time to write the papers and write the grants, then we discuss with the doctors there to make sure that, because, you know, as somebody who has been more involved, let's say, in the technical development, I know a lot more about MR physics than about vascular physiology in the end. You know, we really need to make sure that we talk with the doctors to ensure that the things we're trying to measure and the questions we're trying to ask are the right ones, right? Because we always have our own ideas about this, but if you're not working with the patients, you know, might be completely off base, right? So it's important to always have that dialogue with the clinical people to make sure that we're going in the right direction.
Right. And you've got your students on site doing basically the MRIs, or working with the MRI staff and analyzing the data. So you've had 6 PhD students in the past 5 years, 2 master's students, 2 postdocs. So it's a healthy lab. You published 23 journal articles in the past 5 years, 27 conference publications. I can imagine the constant effervescence, just like so many things to analyze and understand and publish. What do your students bring in this process? I'd like to hear about some of their insights and thoughts as you're working with them, and how have you been surprised, for instance?
Yeah, no, I think, I mean, first of all, working with the students is the best part of this job. And I personally don't think a single individual is creative enough to really figure out those scientific questions, right? It really has to be a group effort where everybody brings their own ideas and their own interest to the question, because there's an infinite number of questions you could ask, an infinite number of things you could do, right? And so it's always a mixture. I'm not a very top-down person, and you know, I always try to make sure that the student is basically driving the question, and I try to set some parameters, of course, in terms of feasibility, but really it's about, you know, a dialogue with the student in trying to figure out what we're going to ask as a question, how we're going to do it. And I think, you know, they're an amazing source of scientific creativity And, that's the best part of this.
Are there any examples that come to mind that, that you'd like to tell us about?
Yeah, so I think, to go back to this exercise study, since I've already talked about it. So this is a poor student who had, she's graduated now, and she did amazing things, but basically everything I tried to set up for her, it just never worked out. The first project, we ran out of money because, we didn't have, the recruitment wasn't working well. And then she was supposed to do something else and a pandemic happened. And so she was very, very unlucky. And so she really had to spend a lot of time kind of bootstrapping her PhD in many ways. And she's the one who really came up with the idea of, taking these kind of exercise questionnaires and really trying to systematically figure out how you could quantify in terms of metabolic equivalent and type of exercise, how you could quantify exercise from these questionnaires, which, don't give you directly that information. Right. And then kind of relating that to brain health and hormones. And, I told her about this data set, but she's basically the one who drove the ideas, right? Okay. And really came up with how we could look at this exercise question in a way that I could never have done, because I actually don't really know anything about exercise. Right,
that's interesting. And was there a temptation then to sort of reach out to applied physiology professors, for instance, or other folks getting engaged? Does that necessarily open up those conversations?
Yeah. So I think, this is a first step. We are just, working on this data set now, but for me, definitely, it opens up a lot of questions that can lead to very interesting collaboration. So for example, I'm starting this collaboration with Marta Kirsten Ertel in engineering to really try to use apps to develop apps to quantify activity and lifestyle a little bit better, and then to use AI to try to look at these big data sets and kind of collect our own data sets for this, right? In a way that, I wouldn't have thought before if we didn't have this project in collaboration with my student.
Right. And what are you hoping for in terms of that collaboration with the AI Institute? What are the potentialities for your research?
Yeah, so I think these are very complex questions, right? These lifestyle, there's so many facets to lifestyle. And I think standard statistical analyses it's difficult to capture what's actually happening, because you have your a priori ideas and perhaps biases about what you're expecting to see, right? And you're trying to fit extremely complex data into relatively simple statistical model that you can come up with as a human person, right? And I think AI has a lot of potential in kind of making us think about these extremely complex data in new ways by extracting patterns that we would have never, from a priori information, come up with. Right. So I think AI is an amazing tool for hypothesis generation. Okay. Basically.
Right. Assuming the data is properly.
Yes, of course. No, data curation is a whole difficult thing to do, but if you do get good data and you have enough of it, then I think AI has amazing potential for generating hypotheses that we would have probably come up with eventually, but who knows how long it would take us to get there.
So it's accelerating the process. It's giving us better tools.
Exactly.
And how do we integrate those hypotheses?
Yeah, I think we don't have a good solution to that yet. I think we're just scratching the surface of what AI can do. But basically, I think what AI can do, at least the way I see it, is help us extract these complex patterns out of the data that are completely based on the data, not on any a priori idea that we had about the data. And then we can look at those, try to see if we can use those patterns that we've extracted to generate new hypotheses about what the physiological process is. And then we can go and find our very quantitative, targeted data to test that new hypothesis about the data.
This is exciting. So you've been at Concordia for 9 years?
Apparently.
And the trajectory is absolutely astounding. And just coming back to your progression from biochemistry to the neurosciences, into physics, and looping into AI, and touching upon cardiovascular and cerebral metabolic imaging. It seems like there's so many possibilities right now, and I can see you involved on so many fronts. What do you imagine for the next 9 years? How do you see yourself? Where do you see yourself going? And what would you love to do, especially?
Well, first of all, I would need an army of students.
Army of students.
And I think really what I'm interested in is trying to really go into these, this direction of trying to understand lifestyle in this very kind of holistic fashion, right? Because I think this very reductionist approach that we've used so far towards understanding lifestyle, I don't think it can work. I think lifestyle has extremely complex effects on, every aspect of the body and mental health. And therefore, we're going to need very different tools than what we've used in the past to try to understand lifestyle. And to be able to do that, we need many more types of data that we've had before. We need, new analysis tools. We need, mostly those two things, I guess. And that's kind of where I want to try to go, is to really explore how we can understand the complexity of it in, using all these new tools, these new data sets, all these new things that are, really starting to emerge now, right? These very large consortia that have these very large data sets, our own, very quantitative data, AI, and, multivariate analysis, which is also, developing really quickly, to really try to understand, how lifestyle in all its complexity can affect, change brain health in all its complexity, right? It's two very complex things kind of, interacting together. And it's very hard to capture that.
And you've cautioned us about taking into account sex and gender, for instance, the different moments in life. When you talk about lifestyle, so I'm coming from the humanities and social sciences, so I might have a different understanding of what you mean. What do you mean by lifestyle data?
Yeah, so lifestyle data can be almost anything that, relates to how a person lives their life. But I think in its most simple form in life sciences, what we're mostly talking about is things like exercise, diet, sleep. Occupation also has a big impact generally. So these kind of aspects, right? And it's actually really hard to quantify them. So things like exercise, to some extent, we know how to quantify that. But how about sedentary behavior? Right? That's been a really expanding topic, but we don't actually have a good way of measuring that. People are not very good at knowing how long they've been sitting. And, does it matter if it's, 10 hours in a row or 5 and 5? Do you do it every day, every other day? Do you... all of these things about sedentary behavior, we don't really know how to quantify that, but even the tip of the iceberg that we can see shows us that it has a huge impact, right? But we don't actually know how to quantify it. And things like, now that everybody has a smart watch and a cell phone, we can probably use that data to quantify it better. But right now, the apps quantify activity, not sedentary behavior. Right. So we would need a way of changing these apps to actually quantify sedentary behavior as well, right? Because probably that also has an impact, right? And then diet, what do? You do questionnaires to people, you get them to take pictures of it, and you analyze it offline using AI, right? There's many different things that we need to be able to quantify to really understand what a person's lifestyle is. And right now, aside from physical activity, and even that, right, we don't have a lot.
Right. And are we being sedentary here now?
Oh, definitely.
Technically, but we're also engaged in conversation.
That's true.
And our brains are imaging up.
Yes.
Is the lifestyle of a professor a healthy one, given what you've been looking at?
That's a good question. I think, it's a desk job often, so maybe not that part. On the other hand, I think, there's a lot of data on how being stimulated, always doing something that takes you out of your comfort zone, that's really good for your brain. And if there's one thing that really keeps you out of your comfort zone is working with students who, always, they're in their kind of, I don't know, brain expansion, phase of life where they're always trying something new and they're interested in lots of things and they can learn things you've never learned about, and that keeps you out of your comfort zone, and that for sure is good for you.
Right, so there's intellectual stimulation, hyperstimulation for the most part, but sedentary
exactly.
Physical existence.
Probably teaching's good for you, you're standing and you're teaching, so that part I would think is good for you. So a mixed bag as usual. It's a bit stressful, so maybe not that.
What gets you up in the morning in terms of research?
Aside from my kids waking me up.
Yes, so that would be the first thing, absolutely.
I think,
after that,
me, I think I'm an understanding junkie, I like, what really gets me up in the morning is there's so many things I'd like to understand, right? It's never ending. And, and not only science, anything. I really love to understand how things work. And that's what gets me up in the morning.
Right.
It's just like a whole day to figure things out.
Nice. And I'm sure that's very contagious enthusiasm as well.
I hope so.
Yeah. Are there topics that you'd love to explore, or some researchers maybe that you've heard about, or streams of research that you're too shy to sort of approach? But can you tell us about that?
So there's a huge thing these days that people are starting to develop imaging techniques for, and that's lymphatic flow. So we know that the blood circulates in the vessels, but there's also liquid outside the cells and outside the vessels. And we're now starting to understand that this liquid often is used in detoxifying the brain, right? Things like those proteins that cause Alzheimer's disease. We know that they actually accumulate in this extracellular fluid and it needs to be drained. And that whole drainage system is so slow that for many, many years, we didn't know if it existed or not. And now we're starting to be able to image that. And to me, that's absolutely fascinating. I'd love to be able to get into that, but I haven't dared yet.
Right. So this is brand new.
Yes.
This is terribly exciting.
Yes.
And who would your interlocutors be there? In what field are they working, and how might you poke in and have a look and collaborate with them?
Yeah, so I think there's the people who develop those techniques. I don't think I would develop my own, right? So kind of the other researchers who work in imaging, and then I think a lot of the topics that are related to this lymphatic flow are some certain diseases like Alzheimer's disease, and then sleep. So what we know is that sleep is when this happens, this flow and this detoxifying, right? So, and I think sleep is an incredibly important part of lifestyle also. Right. So, that would kind of involve going more into the figuring out sleep and how it works and how it interacts with all these other aspects of health and lifestyle to be able to, promote health.
Right, and we know how many people today brag about how little sleep they have. It's a sort of measure of success or importance, and so we might want to reconsider that. You've worked with pre-Alzheimer's patients as well, right? So do you see this study of the detoxifying aspect of the lymphatic system as something potentially interesting? Is it complementary to what you've been doing? Yeah,
exactly. I think it's just another piece of that puzzle of how everything fits together, right? I've been working a lot on vascular health, and there's also indication that the way this flow in the lymphatic system works is because every time there's a cardiac pulsation, it basically pushes the liquid a little bit, and that's what helps to create this flow inside the brain, right? So it's all related together, and that's what's amazing about biology. It's incredibly complex. Everything fits together in this very messy way. And that's what I like about it, is try to understand the whole messiness and complexity of it.
And you're leading us through this complexity. I don't think it's all that messy the way you present it. It's a very coherent, enthusiastic approach to trying to understand overall systems. Claudine, thank you so much for this this conversation.
Thank you. It's always so much fun to talk about these things.
I feel so much smarter now, having listened to you, and I'm very keen to learn more. I'd like to open it up for questions in the audience, if anyone has any or comments online or in the audience.
Yeah, and folks, if you're joining us here in person, just raise a hand, let us know you'd like to speak, and we'll get a mic to you so that we can hear you online. Patrick, I've got no one online raising hands so far, but we invite any chat or audio comments there as well.
We can also have an informal conversation afterwards if this is too much pressure. Good. Great. Well, thank you so much.
Thank you.
And before I conclude, I'd just like to tell people online that this is the same link, if I understand well, for the next conversation with Tianming Zhang at 11 o'clock. So hang on. We'll be back with thoughts on organic contaminants. So thank you so much.
That's right. Thank you.
Yes.
Yeah, and folks, we're actually going to keep the Zoom link open, so you're welcome to hang out in that space for 15 minutes, and we'll be back in 15. Thanks so much for being here.